Welcome back. Now we're going to learn about the small intestine. And when we look at the small intestine, it's about 18 feet in length, and it's called the small intestine because its diameter is smaller than the diameter of the large intestine. They're about this to that. Okay. The small intestine has three different areas. You don't need, the first area is the duodenum, the second jejunum, the third is the ileum, but the duodenum is the one area you really need to learn the name of. So as soon as you're through the pyloric sphincter, you're in the duodenum. Notice it's kind of C-shaped, and it actually dives in the retroperitoneum because it's going to be hooking up with the pancreas, which is a retroperitoneal structure. And then it comes back towards the midline. And as soon as the intestine flips back to come back intraperitoneally, then you're no longer in the duodenum, you're in the second part of the small intestine. And you can't tell from the outside where that part ends and the third part starts, but just be aware that the small intestine ends in the right lower quadrant of the abdomen. Okay. And so the cells in the small intestine secrete a wide range of enzymes, and these enzymes are going to be digesting proteins, carbohydrates, and nucleic acids. Notice I didn't say anything about lipids, and that's because there are no cells in the small intestine that can do anything to lipids. And that's why the small intestine has to hook up to the pancreas. Because that's where it's going to pick up substances that will allow it to not only chemically digest lipids, but also mechanically digest lipids. And so that has to do with the third point here. It's going to receive substances from the accessory organs. And the accessory organs are going to be the liver, which is going to make bile, which is going to travel in ducts, which can go directly into the duodenum, or if it's not needed, for instance, if you're sleeping at night, the liver is still making bile, but the bile would be stored in the gallbladder. And then the pancreas makes enzymes and other substances. And so the bile duct will join up with the pancreatic duct, and they will dump the contents into the duodenum. So all digestion for all proteins, carbs, lipids, and nucleic acids is going to be completed in the small intestines, and all of our nutrients are going to be absorbed in the small intestine as well. And when we look at the small intestine, there are significant modifications to increase the surface area. Even though it's 18 feet long, that still would not be enough length. If we just took that diameter and laid it out flat, it would only be, 3 or 4 inches wide, 18 feet long, not enough to handle all the food and drink that we consume every day. So there are going to be 3 structural modifications we're going to learn about. The first are circular folds. And so circular folds are these things that look like speed bumps. Okay, so there's the cartoon drawing of the speed bump, and here's the actual photograph of a piece of intestine where you can see the circular folds. And they do just what speed bumps do. They slow down how fast the time that has left the stomach can travel through the small intestine. So there's going to be lots of circular folds in the duodenum, and as you move distally, they will decrease in number. The second modification, if you look on the circular fold, you can see these little finger-like projections that are going upward, and each of these finger-like projections is called a villus. And if you look closely, you will notice the villus is just a projection of the mucosa, because here is the muscle layer. So the muscle layer is not going up. We have mucosa going up. It's covered with epithelium, the simple columnar epithelium. We've got individual little goblet cells. And if we look at the villus, we've got a little arterial, a little venous. So we've got a wonderful little capillary bed in the middle of which we have a lymph capillary. And lymph capillaries in the small intestine have a special name. I need you to learn that. It's called a lacteal. It's going to be very important in the small intestine. So that's 2 of the 3 modifications. To see the third modification, we actually need to take a look at an epithelial cell itself. So let's look at one of those columnar cells, and we have a modification we've talked about many times before, and that is we have microvilli. And so when we learned about microvilli on the cell surface, we talked about they're there when cells are going to be either absorbing or secreting. Well, that's exactly what these cells are going to be doing, absorbing and secreting. Okay, so here's a photograph of villi with lots of cells. You can see goblet cells, and if we went in closer, we'd be able to see microvilli as well. So when we take all of this, when we take the small intestine with the circular folds, the villi and the microvilli of this simple columnar epithelium, here's what it would look like if we were looking at it with a scope. Isn't it really kind of cute and feathery? Your small intestine has as much surface area, not 18 feet by 4 inches wide, but a surface area of a tennis court. And that is why this is the major site for digestion and absorption, because there is a huge surface area. Okay, so a couple of more functions. In addition to the usual mechanical digestion associated with peristalsis, the small intestine does a localized churning known as segmentation. So think of it like if you were making brownies and you had the oil and the egg over here and you had the chocolate part over there, and you would mix a little bit this together, and then you mix a little bit this together, and eventually you would have all this homogeneous little structure in little tiny little areas. That is segmentation. Now, talking about absorption, let's go back and look at that villus. What is being absorbed where in that villus? Well, when we look at the arterial and the veins, so stuff has to be absorbed into the cell, specifically then into the blood capillaries, so it can leave through the venous side. What is going to be absorbed into the bloodstream are going to be our simple sugars. So when the carbohydrates are broken down into the simple sugars, like the monosaccharides, so that would be galactose, fructose, and glucose. For this course, you can just remember glucose. So carbohydrates broken down into glucose monomers, although not technically correct, it's good enough for this course. The other thing, when proteins are broken down, they're going to be eventually all broken down into simple amino acids. So you can remember that for this course. I'll be talking more detail later on in a different video about that. So that's what you're going to be finding in the blood here. But there's a couple other things that show up in the blood as well. We have some other things that are required. Most of our vitamins are going to go in our bloodstream, as well as our minerals. So think of things like calcium, phosphorus, magnesium, manganese, iodine. So you can make your thyroid hormone. All of that stuff goes directly into a blood capillary. Now, notice I said most vitamins. So don't memorize the ones that go. It's a very small minority that doesn't go. And those are the ones we will learn. So if when things are being absorbed into the villus, they don't go into the bloodstream, their other option is to go into the lacteal. And that's because they're not going to be water soluble. And so what goes into the lacteal what's going to be in the lymph are going to be our fatty acids. So this is the breakdown products of our lipids, because when lipids are broken down, they're broken down into fatty acids and glycerol. And the other thing are going to be our fat soluble vitamins, vitamins A, D, E, and K. Go in our lymph. And remember, those lymphatic vessels eventually dump back in here where the jugulars meet the subclavian veins. And so they're going to be dumped into your right atrium. So our small intestine has cells that can produce enzymes. So we do have enzymes that help with carbohydrate digestion of some disaccharides. For example, we have maltase, which will break down maltose into a couple of glucoses. We have lactase, which will break down lactose into glucose and galactose. You don't need to know what maltose is made of or lactose is made out of, but I think you can figure out maltase is for maltose and lactase is for lactose. Then we also have some proteases for protein digestion. In this case, it's called a peptidase because it's not going to work on a big protein. Remember, we already have in our stomach, we already took our protein and broke it down into peptides. So now we have a peptidase, which is going to take that peptide and break it down into amino acids. And our small intestine also makes something called a nucleosidase, which takes a nucleoside and will break it down into a base and a sugar. And I'll explain that more in that video where I talk about digestion and absorption some more. So that's it for the small intestine. I will see you in the next video where we're going to talk about 3 more accessory organs, the ones that are found in the abdominal cavity. See you shortly.